Hello, my name is Sabrina Simoncelli, and I am one of the newest group leaders within the London Center for Nanotechnology and the Department of Chemistry within UCL. So in my group, we use and develop nanoscale technologies to image, sense, and manipulate biological objects. Currently, we are interested in understanding how the spatiotemporal organization of proteins in immune cells allow them to combat diseases and maintain health. One of the most important players in our immune systems are a type of white blood cell, which is known as the T lymphocyte or T cells. Now, T cells patrol our body in search for intruder, and they trigger an immune response when they found them. To launch this immune attack, T cells recognize foreign substances, which are known as antigens, via receptors on their surface. After this receptor antigen binding, a cascade of chemical reactions instructs the cell on how to behave. And the molecular machines that control these biological processes are called proteins, each with a specific task and location. Some of the most powerful signaling molecules in T cells are actually transmembrane proteins. These are proteins that are embedded in the membrane and that can protrude above the surface to detect antigens. Now, cytosolic proteins are also very important because they work by interacting with transmembrane proteins at a specific time points after T cell activation in order to fine-tune immune responses. In our group, we are interested in understanding the critical steps that result in T cell activation, such as, for example, the formation of nanoscale protein clusters, which mechanisms are in place in order to prevent aberrant activation, and whether we can identify optimal design sweet spots to engineer nanobacteria that can eventually modulate immune responses. Now, this is important because to prevent aberrant activity that could eventually provoke cancer or autoimmune diseases, the activation and functioning of T cells has to be tightly regulated. So a detailed molecular descriptions of the events that underlie T cell function is of immense value to design novel therapeutics. Unfortunately, there are several technological limitations in detecting these rearrangements of T cell signaling proteins and these nanoscale clustering domains. Proteins are 1,000 times smaller than a cell. A cell is already 1,000 millimeters in size. So to see single proteins in action interacting in and out the membrane area, we need to use sophisticated light microscopes that allow us to resolve features in the nanoscale domain. This is why advances in microscopy in general and cell biology are usually intimately intertwined. Now, fluorescence microscopy is one of the most widely used tools in biological research, and this is because of its specificity, high sensitivity, and low invasiveness. However, because of the intrinsic properties of light, standard fluorescence microscopes can at best resolve features which are around 200 nanometers apart. During the last 20 years, there has been a lot of technological innovation, including the development of super resolution fluorescence microscopy techniques. And this has allowed us to improve the resolution that can be achieved with an optical microscope. In the lab, we use a combination of experimental and computational techniques, which include optical microscopy, nanofabrication, spatial statistics, and image processing in order to improve current super resolution imaging technologies. We recently have developed methods that allow us to resolve features with sub-10 nanometer resolution in 3 dimensions. And at present, we are exploiting this 3D nanoscale imaging technology in order to learn more about specific molecular pathways involved in the immunological synapse.